Section A. 1a. From Figure 2.1, identify the substances which enters the plant by 1. Osmosis. Correct answer, water. 2. Active transport. Correct answer, nitrates potassium, except either or both. 3. Diffusion. Correct answer, oxygen. B. Figure 2.2 is a graph showing the effect of pH on the rate of enzyme-controlled reactions N and O. 1. What term is given to the parts labeled X and Y in the graph? Correct answer, optimum pH. Acceptable answers, maximum rate of reaction to optimum point. 2. Which reaction is under acidic conditions? Correct answer, reaction N, since its curve and peak fall between pH 1 and 7. 3. Which reaction could only be for a protein food? Give a reason for your answer. Reaction N. Reason, reaction N has an optimum pH of 4, which represents an acidic medium. This matches the conditions required by standard protein-digesting enzymes, like pepsin in the stomach, to function efficiently. C. Explain the role of enzymes in metabolism. Correct answer, enzymes function as biological catalysts that speed up the rate of chemical reactions in living cells by lowering the activation energy required for reactions to occur. They regulate all metabolic processes without being altered or consumed themselves. This includes controlling catabolic reactions, breaking down larger molecules to release energy, such as in respiration or digestion, and anabolic reactions, building up complex molecules, such as protein synthesis. 1a. Movement of substances into plants. Osmosis. Water osmosis is a unique type of diffusion specifically involving water molecules. Soil water has a higher water potential than the cytoplasm of the root hair cells. Because the root hair cell membrane is partially, selectively, permeable, water naturally moves down its water potential gradient into the plant without using cellular energy. Active transport, nitrates and potassium plants require mineral ions like nitrates for making amino acids or proteins, and potassium for enzyme activation and cell turgor. Often, the concentration of these ions is already higher inside the plant roots than in the surrounding soil water. To pull them in against this concentration gradient, the plant must use energy in the form of ATP to pump them across the membrane. Diffusion, oxygen root hair cells are living cells that undergo aerobic respiration to generate the energy needed for active transport. They obtain the required dissolved oxygen directly from air pockets in the soil. Because oxygen is constantly being consumed inside the root cells, a steep concentration gradient exists, allowing oxygen to passively diffuse into the roots from high to low concentration. 1b. Enzyme activity and pH, Figure 2.2. Optimum pH at points X and Y. Every enzyme has an ideal environment where its three-dimensional shape, conformation, is perfectly stable, allowing its active site to bind perfectly with its substrate. Points X, pH 4, and Y, pH 11, mark these peaks. If the pH moves too far below or above these points, the excess H+ or OH ions disrupt the ionic bonds holding the enzyme's structure together, causing it to denature and lose function. Acidic conditions, reaction N. The pH scale ranges from 0 to 14, where 7 is neutral, anything below 7 is acidic, and anything above 7 is alkaline. Looking at the curve for reaction N, it begins to function at pH 1, reaches its maximum efficiency at pH 4, and drops off near pH 8. Because the vast majority of its activity occurs below pH 7, it is classified as operating under acidic conditions. Protein foods and reaction N. In animal physiology, the most notable site of highly acidic enzyme action is the stomach, which secretes hydrochloric acid. The primary enzymes active here are proteases, such as pepsin, which break down long protein chains into smaller polypeptides. Since reaction N thrives at an acidic pH of 4, it mirrors the behavior of these stomach proteases. 1c. The role of enzymes in metabolism. Metabolism is the sum of all chemical reactions taking place within a living organism. These reactions generally happen too slowly at normal body temperatures to sustain life. Enzymes solve this problem by acting as biological catalysts. They possess a specific region called an active site that binds to substrate molecules. By holding the substrates in a precise orientation or putting stress on their chemical bonds, enzymes significantly lower the activation energy, the minimum energy required for a chemical reaction to start. This catalysis allows the body to seamlessly balance its two metabolic pathways. Anabolism, building up synthesizing large molecules, such as linking amino acids together to form proteins, or combining carbon dioxide and water to make glucose during photosynthesis. Catabolism, breaking down splitting large molecules into smaller units, such as breaking down starch into maltose during digestion, or converting glucose into carbon dioxide and water to release energy during cellular respiration. 2a. Enzyme definition and optimum temperature. 1. What is an enzyme? Correct answer: a biological catalyst that speeds up the rate of chemical reactions in living organisms by lowering the activation energy, without being used up or altered in the process. Acceptable answers: an organic catalyst, a protein that speeds up chemical reactions in cells. Further explanation: enzymes are made of proteins folded into specific three-dimensional shapes. They have an active site where substrates bind. They are vital because, without them, the chemical reactions required to sustain life would happen far too slowly at normal body temperatures. 2. What is the optimum temperature of the enzyme reaction shown in the graph in Figure 2.0? Correct answer: 37:/circ/text C, except variations between 37:/circ/text C and 38:/circ/text C based on reading the peak line. Further explanation: the optimum temperature is the specific temperature at which an enzyme works at its maximum possible rate. On a graph, this is always found by tracing down to the x-axis from the absolute peak of the curve. At this point, kinetic energy is high enough to cause frequent collisions between enzymes and substrates, but not high enough to disrupt the enzyme's structure. 2b. Enzyme denaturation. 1. At what temperature in Figure 2.0 is the enzyme completely denatured? Correct answer: 60:/circ/text C. Acceptable answers: any value from 58:/circ/text C to 60:/circ/text C, where the curve completely flattens out at the bottom of the temperature scale. 2. Give a reason for your answer in B, I above. Correct answer: at 60:/circ/text C, the rate of reaction drops to 0, indicating that the enzyme's active site has permanently changed shape and can no longer bind to the substrate. Acceptable answers: the curve hits the x-axis, the reaction has completely stopped. Further explanation: as temperature rises past the optimum point, the intense thermal energy causes the atoms within the enzyme protein to vibrate violently. This breaks the delicate hydrogen and ionic bonds maintaining the enzyme's specific 3D shape. Once these bonds break, the active site loses its shape permanently, denaturation. Even if the temperature is lowered later, the enzyme cannot regain its shape, meaning substrate molecules can no longer fit into the active site, the lock and key mechanism fails, and the reaction stops completely. 2c. Temperature regulation in medicine and health. 1. Explain why it is important to measure the body temperature when a person is sick. Correct answer: to monitor if the body temperature has risen above normal, fever, or dropped too low, which can dangerously affect enzyme activity and metabolic functions. Acceptable answers: to check for a fever to ensure body temperatures do not reach levels that can denature vital metabolic enzymes. Further explanation: human enzymes are optimized to function perfectly at normal body temperature, around 37:/circ/text C. When a person is sick, a fever is often triggered by the immune system to fight infection. However, if the body temperature rises too high, e.g., above 40:/circ/text C, vital metabolic enzymes in organs like the brain can begin to denature, leading to cellular damage, seizures, or organ failure. Measuring temperature helps determine if medical intervention is needed to protect these enzymes. 2. Propose 2 ways the body temperature of a sick person can be brought down to normal. Way 1. Administering antipyretic medications, such as paracetamol, aspirin, or ibuprofen. Way 2. Physical cooling methods, such as wiping the body with a damp cloth, tepid sponging, or removing excess clothing or blankets. Acceptable answers: drinking plenty of cool fluids or using a fan for convective cooling. Further explanation: antipyretic drugs work chemically by acting on the hypothalamus, the brain's thermostat, to reset the body's target temperature back to normal. Tepid sponging utilizes physical principles of thermodynamics. As the water on the damp cloth evaporates from the warm skin, it absorbs and carries away latent heat from the body, lowering the surface temperature safely through evaporative cooling. Using ice-cold water should be avoided as it can cause shivering, which paradoxically raises internal core temperature. 3a. Term for points J and L. Correct answer: optimum pH. Acceptable answers: optimum point, maximum pH activity point. Further explanation: the peaks J, at pH 2.5, and L, at pH 11.2, represent the specific hydrogen ion concentrations where the respective enzymes have their highest catalytic activity. At these points, the chemical bonds stabilizing the enzyme's tertiary structure are perfectly balanced, rendering the active site fully functional and complementary to the substrate. 3b. Drop in reaction rate after points J and L. Correct answer: the rate goes down because the enzymes become denatured. Moving away from the optimum pH alters the ionic charges on the amino acids making up the enzyme, which breaks the chemical bonds that maintain its specific three-dimensional shape. This changes the shape of the active site, preventing the substrate from fitting. Acceptable answers: extreme pH changes cause the enzymes to denature. The enzyme's active site loses its shape, so substrate complexes can no longer form. Further explanation: enzymes are sensitive to the concentration of H+ and OH- ions in their environment. If the pH increases or decreases past the optimum point, these excess charges disrupt the hydrogen and ionic bonds holding the protein structure together. Once the active site is distorted, denatured, the lock and key mechanism fails entirely, bringing the reaction rate down to 0. 3c. Regions of the alimentary canal. Reaction K, stomach. Reaction M, small intestine, duodenum, ileum. Further explanation: reaction K peaks in a highly acidic zone, /approx /text, pH 2.5. This perfectly mirrors the environment of the mammalian stomach, which secretes hydrochloric acid, /text, HCl, to destroy pathogens and activate gastric enzymes. Reaction M operates optimally in a strongly alkaline environment, /approx /text, pH at 11.2. In the human alimentary canal, alkaline conditions are established in the small intestine when acidic chyme from the stomach is neutralized by bile and pancreatic juice, which contains sodium hydrogen carbonate. Note: while human intestinal enzymes typically peak closer to pH 7 to 8, in standard examination contexts featuring an extreme alkaline curve, the small intestine remains the expected physiological region. 3d. Specific enzymes and nutrients. 1. Suggest one enzyme which can catalyze reaction M. Correct answer: trypsin, pancreatic amylase, pancreatic lipase. Acceptable answers: maltase, peptidase, sucrase. Further explanation: these are all enzymes found in the pancreatic juice or intestinal juice that function within the alkaline medium of the small intestine. 2. State one food nutrient which can be catalyzed in reaction K. Correct answer: protein. Acceptable answers: proteins or polypeptids. Further explanation: the stomach primarily digests proteins. The main enzyme active in this region is pepsin, a protease that breaks down complex protein chains into simpler peptides under strongly acidic conditions. 3e. Other factors affecting enzyme activity. Factor 1. Temperature. Factor 2. Enzyme concentration to substrate concentration. Acceptable answers: presence of inhibitors, presence of coenzymes or activators. Further explanation: temperature. Low temperatures slow down molecular movement, low kinetic energy, reducing collisions between enzymes and substrates. High temperatures past the optimum permanently denature the enzyme. Substrate and enzyme concentration. increasing the amount of substrate or enzyme molecules increases the frequency of successful active site collisions, accelerating the reaction rate until a saturation point is met. AI. What is the general name given to a biological catalyst? Correct answer: enzyme. Acceptable answers: enzymes. 2. Give a reason for boiling the potato discs in experiment 2. Correct answer: to denature the enzyme, catalase, present inside the potato cells. Acceptable answers: to destroy or inactivate the biological catalyst using high heat. 3. Explain the difference in results obtained between experiments 1 and 3. Correct answer: experiment 1 lacks a catalyst, 0 potato discs, so hydrogen peroxide decomposes at a very slow, natural rate, producing only 1.5 of oxygen. Experiment 3 contains raw potato discs which provide the active enzyme catalase. This enzyme significantly speeds up the breakdown of hydrogen peroxide into water and oxygen, yielding a much higher volume of oxygen 10. Acceptable answers: experiment 3 has active enzymes that accelerate the reaction, while experiment 1 has no enzyme, resulting in a slow spontaneous reaction. B. Predict what would be the result of 1. Increasing the number of potato discs in 5\text cm(3) of hydrogen peroxide. Correct answer: the rate of reaction will increase, causing oxygen to be produced much faster. A higher volume of oxygen will be collected in a shorter period. Acceptable answers: more gas bubbles, oxygen will be produced quickly. Note: the final total volume of oxygen depends on the limiting reactant hydrogen peroxide, but the initial rate of production will definitely jump. 2. Heating the mixture of 5 of hydrogen peroxide with 5 potato discs. Correct answer: if heated moderately up to the optimum, the rate of oxygen production will increase. If heated to extreme temperatures above 60, the volume of oxygen will drop back down to 1.5. Because the enzymes will denature. Acceptable answers: the reaction will speed up initially and then stop completely if the temperature gets too high due to denaturation.